Okay, blood part 2. Now we need to talk about how a multipotent, uncommitted hematopoietic stem cell differentiates into red blood cells, platelets, macrophages, neutrophils, eosinophils, basophils, and B and T lymphocytes. So the undifferentiated stem cell has many potential different fates. So let's start at the beginning. We have our stem cell up here. This is our multipotent uncommitted hematopoietic stem cell right here. That's where it all begins. And this then travels down this road to become a committed stem cell. That means it's been given its assignment. It knows what to differentiate into at that point. Let's start off over here and travel down to this side first. So it can, the stem cell can be committed, and it can come from the bone marrow right here, and then essentially be what we would say is a lymphocyte precursor. A little rain our room a little bit. And this can differentiate into one of two things. A B or a T lymphocyte. And we'll get to, when we get to down here, we'll talk about the differences between all of these. So that's one possible fate. The stem cell could be turn into a lymphocyte precursor and then eventually differentiate completely into a lymphocyte, a B or a T lymphocyte used in our immune response. We also have plenty of other different responses that we can do. Okay. So we'll start here and we'll work our way to the right. So of our first stop that we can do, we can get this, we can start differentiating into what's eventually going to end up being a red blood cell. So we have these like such. The next step along the pathway is a reticulocyte. Kind of looks like that, and then eventually get to our red blood cell, which is a biconcave disc down here. The next one differentiates kind of similar to what we have here, and then we get this kind of weird shaped thing here that looks like that. Got an internal structure such like that. And then this differentiates into platelets. Remember, platelets are just cell fragments that float around in the blood. Next we get our blast cells. And our blast cell has two potential fates. My arrows are going to have to move along a little bit. So first stop in our differentiation to their next stop, which is a monocyte, which differentiates to a tissue macrophage. Remember, tissue macrophages are going to clean up all the garbage in the cell. They're going to eat it up. Our other fate here is something like this. And this is called a promyelocyte, which differentiates into our next step called a band. To a neutrophil. The next one is, I'm going to come around this way and make us some space right here. This differentiates into more of a blob here, and then finally comes down to this shape, which is an eosinophil. And lastly, on this side, I'm going to come over here. We have this cell. And it's going to, I'm just going to follow it right over to here. I'm going to give you something like this. And it's going to differentiate into, over here, we call it basophil. So now that we've drawn them all out, it's important to talk about some of these things. So let's talk first. I tell you what, let's stop here, and then we'll start on the next video.